Hi, this is Steve St. John, Professor of Psychology at Rollins College. This is the second in a series of videos on sheep brain dissection. In this video, we'll be looking at structures that you can see on the ventral surface or bottom side of the whole sheep brain. Okay, so here we have a whole sheep brain, but now it's dorsal side up, so I'm just going to flip it over so that it is ventral side up so that we can look at some of the most notable features on the ventral surface of the sheep brain. And I'm going to work my way from anterior to posterior and point out some of the larger structures that can be seen. So the first clearly identifiable structure and one of the most anterior structures in the central nervous system are these two parts of the brain that seem to not be attached except at their base. And those structures are called the olfactory bulbs. They're the first part of the central nervous system that receive input from the olfactory receptors of the nose. So they're part of your sense of smell. Posterior to the olfactory bulb, there's a very prominent white-shaped structure here. This would in life be attached to the eyes, and this is the optic nerve. Depending on the dissection of the removal of the brain from the skull and the removal of the brain from the meninges that wrap around the brain, you might get a very small piece of optic nerve. In this case, we have a really nice long portion of optic nerve. And it's very white in color too, which distinguishes it from the rest of the brain. You also notice that the optic nerves seem to travel over one another and back into the brain, forming an X. And in fact, early anatomists were impressed with this X and called it the optic chiasm. And chiasm, basically a word that means crossing or X shape. And so that whole structure there, the optic nerves, feed into the optic chiasm. In reality, only some of the axons from this would be the left eye cross into the right side of the brain. Some of them, half of them, will stay on the same side, but it does form this X-like structure on the ventral surface of the brain that's right beneath the hypothalamus. Posterior to the optic chiasm, you can see what looks like a slightly pinker or browner colored shape. Whenever you see sort of a brown color, it typically indicates that it's a structure that's got a lot of blood vessels in it. And indeed, this is part of the pituitary gland, which sits just under the hypothalamus and just behind the optic chiasm. So it's part of your endocrine system, but is intimately associated with the central nervous system. Sort of left and right, or lateral to the pituitary gland, you can see a couple of prominent blood vessels and where they bring the blood supply into the brain. Very lateral to the pituitary gland, you can see a little bit of cerebral cortex that is coming around to the underside of the brain. And I mentioned in previous video on dorsal structures that you could see 3 of the 4 lobes of the sheep brain from the top surface. This is the 4th lobe, which in humans would be called the temporal lobe. In sheep, it's often called the piriform lobe, and it's somewhat similar to the temporal lobe in humans, but much, much smaller. Going back from the pituitary glands, if you have a careful dissection of the dura mater, the meninges that overlie the brain, you can often extract another prominent nerve, which also goes to the eye. This is cranial nerve 3, the oculomotor nerve, and it controls some movements of eye muscles and also some pupillary reflexes of the eye. The oculomotor nerve emerges from the midbrain. So the optic nerve emerges from the forebrain. So somewhere in between here and here, we're entering the bottom surface of the midbrain. Forebrain, midbrain, and hindbrain. As you move the probe back, you might not be able to see the 3-dimensional structure here, but we actually reach kind of a little bump or a little hill here as we leave the midbrain and enter the hindbrain. And that bump is called the pons. Pons kind of resembles an Adam's apple on the neck of the brain, if you will. You can see a little bit of the lateral hemispheres of the cerebellum. because we're now back in the hindbrain, and the cerebellum sits on top of the pons in the structure behind the pons, this large structure, which is the medulla oblongata. And then, again, if you just lightly bring your probe back, it'll settle into another groove, and that's more or less the beginning of the spinal cord. Okay. You may be able to identify some smaller nerves. There are 12 nerves that emerge off of the brain. They're numbered from 1 to 12 from front to back. The first one is the olfactory nerve, which you usually can't see because it's a bunch of little fibers that run through holes in the skull. But the optic nerve is the largest, and that one's very prominent. It's the largest in the sheep. Then cranial nerve 3 is the oculomotor nerve. Cranial nerve 4 actually emerges from kind of the side, and sometimes you can see that because it's kind of protected where it emerges from the side here between the cerebellum and the cerebrum, also emerging from the midbrain, also a nerve involved in eye muscles, eye movements. There's a nerve here, and then you may see, especially sort of emerging from the side of the medulla, other nerves or complexes of nerves, which are among the remaining cranial nerves. But they can be a little bit difficult to identify because it's sort of hard to remove them intact in a way that you can match up easily with a dissection guide or pictures that you might be looking at. So, quick little review from front to back. We have the olfactory bulbs, we have the optic nerve and optic chiasm, we have the pituitary gland, the piriform lobe, the oculomotor nerve, the pons, the medulla, and the spinal cord.